Good day, gentlemen. Today is Wednesday, April the 23rd. No, I lied. Yeah, Wednesday, April the 22nd. All right. And today we are going to start chapter 16, which is the molecular basis of inheritance. Now, we started the other day and we talked in chapter 15 about the idea that genes were being passed on from one organism to its offspring. And we're going to continue that discussion today, but here's the thing. What you have to understand is that in the early days, in the 1800s and the early 1900s, they kind of knew what genes were. Remember, Mendel called them factors, but we didn't know chemically what those factors actually were or how they were physically passed on from a sperm and an egg or into a sperm and an egg and then onto the zygote. So the story begins, actually, we'll kind of cut in halfway through the story. In 1953, James Watson and Francis Crick introduced their model of DNA. It was a double helix. And essentially what that is if you take a long extension ladder and you grab it at the top and the bottom and you twist, all right, that's essentially what a double helix actually looks like. We know today that the DNA is the hereditary molecule, and it's reproduced in every single cell in your body, at least in some point during the developmental process. And the DNA is what controls pretty much every aspect of your physical being, your biochemistry, your anatomy, your physiology, and in some cases, even your behavior. All right. This is just a very cool image of DNA molecules. And DNA is copied during replication. And cells with damaged DNA can repair that DNA. We'll get back to that later on in this chapter. So, section 16.1 is dealing with the concept that DNA is the genetic material. All right, and so remember, again, going back to the early 1900s, all right, Mendel had identified this idea of factors, but we didn't know what molecule was the factor of inheritance. And so this was one of the big questions of the early 1900s, at least in terms of biology. It kind of began with Morgan, the guy who studied fruit flies. Remember him from chapter 15. And he was able to show that genes were located on chromosomes. But there were two different components in chromosomes. Chromosomes are made not only of DNA, all right? That's kind of a myth that's out there. It's not just DNA that makes up chromosomes, but also proteins that hold the DNA together. And so both of them, DNA and protein, became candidates for genetic material. And it wasn't until people started studying bacteria and viruses that DNA really became the front runner for the molecule of inheritance. So in 1928, a guy by the name of Joe Griffith did an experiment with two different strains of bacteria. They were very closely related, but one of them was pathogenic and the other one was harmless. So when he mixed heat-killed bacteria with living bacteria of the harmless strain, the living cells became pathogenic. So what that suggested was that somehow those living cells were taking the DNA from the dead cells and incorporating it into themselves. All right. Griffith called this whole process transformation. And it's now defined as a change in not only the genotype, but also of the phenotype of a cell because of the assimilation of DNA that came from somewhere else. This is a nice little summary of his experiment. So he started out with living S cells. The S stands for smooth because they're covered in this coat that made them gave them a smooth appearance. But this is a pathogen. And when these S cells were injected into mice, the mice died. Bye-bye now. He then took living R cells. R stands for rough because they have this rough exterior. And he injected them into mice, and the mice survived. Yay! Nice little micey. Then he took heat-killed S cells, all right, dead cells now, and injected them into mice, and found that the dead bacterial cells didn't cause any disease in the mice. But when he took a mixture of heat-killed S cells and average American R cells, the mice died again. Okay, and again, that indicates that the DNA from the smooth cells somehow ended up in the rough cells. Now, other scientists that were involved in this, Avery, McCarty, and McLeod, identified the transforming substance as DNA. But because the structure of DNA was still not known, a lot of people remained skeptical. So, where does this all bring us? So, more evidence for DNA as the genetic material came from studying bacteria and viruses. So, the brown on this slide represents the plasma membrane of a bacterial cell, and this thing is...
It's 7 o'clock.
Thank you, Jesus. Is a funky little virus, all right? Here's the phage head. The DNA is located here inside the phage. It's got a tail and then a fiber that actually has the ability to inject the DNA right into the bacterial. And you can see some of that genetic material being inserted here. These viruses are called bacteriophages because they infect bacteria. All right. Now, a virus is mostly DNA, sometimes made of RNA, and it's enclosed in a protective protein coat. So all this stuff you see over here in green is protein. All right. I'm going to upload this PowerPoint to the portal so that you can go back and watch this video. It's a cute little video. Take the time to have a look at it, but I don't want to waste time in my YouTube thing here doing that right now. All right, so the next step, now 20-ish years after Griffith's experiment, Hershey and Chase showed that DNA is the genetic material of a virus, a phage, known as T2. They designed an experiment that showed that only one of two components of T2, either DNA or protein, enters an E. coli during infection. And they've concluded that it was the injected DNA of the phage that provides the genetic information. Now that experiment is shown here, all right? Two different steps here. I'll put myself right in the middle. So the first batch had radioactive sulfur, and sulfur is only found in proteins, all right? And so they gave these phages radioactive protein. They allowed the phages to then infect bacteria. They then put them in a blender and shook them up, which shook off the outer coats here. And then they centrifuged it and formed a liquid and a pellet. Now the cells formed the pellet, okay? And what they found was that there was no radioactivity in the cells, but there was a lot of radioactivity in the pellet. So that indicates or suggests that the radioactivity stayed with the proteins, okay? Now in batch 2, they did kind of the same thing, but this time they put radioactive phosphorus in DNA. Same basic procedure. They allowed the phage to infect the bacteria, then they stirred them up to release the protein coats and centrifuged them down. This time they found no radioactivity in the protein, in the liquid portion, but they found come on. They found radioactivity inside the pellets, therefore meaning that the cells had been infected. So that indicated that it was the DNA in these viral bacteriophages that was infecting the bacteria. Again, another little animation of the Hershey Chase experiment. Worth your time to go through the PowerPoint and have a look at this. Please take the time to do it. Okay, now I've cut this video in half because I was afraid it was going to be too long. I think it probably will be. So there's actually going to be two videos today, gentlemen. So two. So please watch the next one, and I will see you in a couple of minutes on the other side.